can hear me very well?
Can.
Okay. Hi everyone. Yeah, I'm just gonna be a host for our topic CME today, as you have been know. So thank you for giving me and Ang Peng Peng the opportunity to present two new topics, which is updates on vaccine, COVID vaccine, and later on I will be giving some updates regarding some of the so-called novel therapy in COVID-19, whereby there's been some updates for the past 1 month, mainly on the monoclonal antibodies and the JAK inhibitors. So if everyone is ready, then I will just probably let Ang Peng Peng to start first.
Okay. I'll share my screen. Okay, good afternoon, everybody. I'm Ang here from Penang GH. So the topic given today is update in COVID-19 vaccine. Everybody can hear me?
Can.
Okay. So we all know that this current COVID-19 ongoing outbreak is unlikely to end in a very near future. Of course, eventually this outbreak will be end, but it will have to cause a lot of death before this outbreak ends. So there is no effective treatment up to now, and we are still not too sure about the disease. So looks like vaccination will be the only effective strategies to help to end the outbreak. The epidemiologists have come out with an analysis and saying that in order to end the pandemic for this COVID-19, at least 70% of the world population need to be immune, either by a natural infection or by vaccination. So if my calculation is correct, at this moment, actually the total world infected population is actually less than 1%. If we want to achieve 70%, either many people need to die first, or either we really have to come out with an effective vaccination program. So we know that a lot of researchers and vaccine candidates are really working hard to look for a solution, but just not too sure that when we talk about vaccine, we are actually working hard, or is vaccine hardly work. Why do I say that? Actually, a lot of vaccine rarely reach the public even until after the outbreak. A very good example is our Ebola disease. Ebola outbreak has been over for 5 years, and the vaccine only available early this year. Then we look at our other outbreak disease like SARS, MERS-CoV, and Zika. Even until now, we still don't have any vaccine for this disease. But of course, the previous effort won't be wasted, because whatever have been developed and studied for those SARS, MERS, and Zika actually assisting the current development of the COVID-19 vaccine. This diagram just to illustrate to you guys that how quickly our COVID-19 vaccine development. If you look at when we compare to the previous infectious disease outbreak like Zika, Ebola, MERS, SARS, even after a year, most of them are still in a preclinical development or clinical trials in Ebola. But if you look at COVID-19, about 2 months after the pandemic being announced, the animal study and human study has already set in. It's really fast and really rapid. Traditionally, if we want to develop an effective and safe vaccine, we need to go through a very lengthy steps and trials, go through animals trials, then human trials, and usually it should take up more than 15 to 20 years. So now we are trying to come up with a COVID-19 vaccine in less than a year. So the concern about the safetyness and the effectiveness of the vaccine is definitely a big issue. So how close are we now to a COVID-19 vaccine? So up to date, there are 212 vaccine candidates been studying. And out of these 212 vaccine candidates, 164 are still in the preclinical, still in the animal or in the laboratory level, and 48 are clinical, and 11 already in the phase 3. In order to design a vaccine, first of all, the researchers need to identify which antigen will be the most appropriate and which antigen is the most suitable one to mount the appropriate immune response in a human's body. Then after identify the appropriate antigen, then we'll continue on to choose the appropriate vaccine platform. Then after that, through the animals test and human test to choose the appropriate routes and regimens for regime. I think everybody is familiar with this structure of coronavirus. This SARS coronavirus 2 consists of 4 important protein, which are membrane protein, envelope protein, nucleocapsid protein, and spike glycoprotein. So this S protein has been recognized to be the most important and the antigen in this coronavirus that can induce a strong antibodies immunity in a human host. So all the vaccine candidates are targeted into producing these S protein antibodies. And this S protein antibody is the only neutralizing antibodies that could be found in this disease. How about N protein? When the researchers study in this N protein in animal levels, it shows that N protein do not have any protection. And worst case scenario that actually N protein could cause a ADE, antibody dependence enhancement disease. And membrane protein and envelope protein both are low immunogenicity. They can't really induce a strong immunity to protect us from getting a disease. So looks like everybody is looking at the S protein to develop a vaccine. So now we have recognized that S protein is the most important antigen. So now look at how scientists, researchers choose the vaccine platform. Generally, there is 5 vaccine platforms that researchers use to develop COVID-19 vaccine. They are viral vector vaccine, protein subunit vaccine, live attenuated virus vaccine, inactivated virus vaccine, and nucleic acid vaccine, which is our messenger RNA or our DNA vaccine. So this is a few examples to show you that the existing vaccine that using a different kind of vaccine platform. So regardless what kind of vaccine platform that researchers trying to develop into this COVID-19 vaccine, eventually is hoping that eventually we are hoping that after the vaccine go into our body, we can come out, we can produce a spike protein and induce a strong immunity response, and then we get protected by the vaccine. So I make this, I put all these 5 different vaccine platforms into this table so that we can have a very clear we can see the difference quickly. Okay, first I would like to talk about this nucleic acid vaccine, which are DNA and mRNA. In fact, this vaccine technique is really new, and we in the world now we don't have any vaccine that actually was produced using this platform. Okay, so instead of using DNA and messenger RNA will be the preferred one, because if they use the DNA-based vaccine, the vaccine when go inside our body, they need to go through the nucleus membrane in order to train our cells to produce the S protein. But if they use a messenger RNA, then they can skip this step. So postulate that this messenger RNA vaccine can induce immunity faster than DNA. So this technique can develop really quickly and can easily manufacture and can produce massively, and is really suitable for a outbreak situation and pandemic situation. And another good thing about this nucleic acid vaccine is do not contain any live segment of the virus, so it do not cause a COVID-19 disease. But because generally, if your vaccine platform didn't use any live component, the immunity that induced will be quite poor, and it usually require multiple doses. But because this nucleic acid vaccine is a new technology, and anticipate that it need to go through a very long and lengthy regulatory approval period. So example of a vaccine candidate that currently in phase 3 clinical trials that very popular is this Moderna and Pfizer, which two of them are already midway of the phase 3 clinical trials. In fact, they already come out with their prelim analysis data and claim themselves are more than 90% efficacy. So the second vaccine platform I would like to talk to is vector-based vaccine. So what happened is the researchers will use pseudovirus. Usually they will choose the animal virus, meaning the virus which won't cause a disease in the human body. So adenovirus and a vesicular stomatitis virus are the most popular virus in using this technique. So they will replace one of the genetic material of this virus with COVID-19 S protein genetic material. So after injecting this vector-based vaccine, then the virus will replicate inside our body and produce more and more S protein, and the antibodies will be produced. So this is how it should work. So this kind of platform is good because when the pseudovirus will be able to replicate and produce the whole length of the antigens or protein that we want. And usually the immunity that being induced are good for a long-term protection. But of course, everything comes with disadvantages. Using this technique is really not too cost effective, especially during in this pandemic situation. And if a patient who have a pre-existing immunity towards the virus, let's say adenovirus, then the effectiveness will be reduced. But this could be avoided because the scientists are actually using the animal form of adenovirus. So there are currently 4 candidates, vaccine candidates are developing vaccine using this technique. Very well-known one is this collaboration University of Oxford with AstraZeneca, Janssen, and the China, one of the vaccine candidate from China and a Russian, and both of them are already started using in the military field. The third platform is inactivated vaccine. They're using the real COVID-19 virus, but inactivated it chemically. So basically the vaccine, the virus is dead. So it can be used in immunocompromised people. But again, like what I mentioned just now, as long as there is no live components of the real virus, usually the immunity is low and they cannot induce a very strong immunity in the host body. So it will usually require multiple doses and also require adjuvant. 2 examples of vaccine candidate are currently in the phase 3 clinical trials are Sinovac and Sinopharm. And Sinovac, although already started using in the China military, but recently it has been suspended in Brazil because there is 1 case mortality after vaccinated. So next platform is attenuated live vaccine. So as the name apply, you understand, very good example is our chickenpox vaccine. So this is really use a real virus that we weaken it, all right? So this will be the best vaccine, whatever disease or whatever vaccine you want to develop, this technique will be the best. But unfortunately, live attenuated vaccine is not suitable for a disease that you don't really understand and for a disease that lacking of effective treatment, because there is small risk that, very small risk that this live attenuated vaccine can become reversal and causing a real disease, and is not suitable for an immunocompromised patient. So until now, there is none of the attenuated live vaccine candidate have reached phase 3 clinical trials for COVID-19 vaccine. So the last vaccine platform I want to talk about is protein subunit. So like what I mentioned just now, we have already identified that S-glycoprotein is the most important antigen to develop into this vaccine. So the researchers could actually purify this S protein and make it into a concentrated material and vaccinate, become a vaccine. And because we can choose the specific antigen that we want, and the benefit is without any other antigen that could cause antibody dependent enhancement disease. So this protein subunit vaccine is actually quite safe, and it also can be used in the immunocompromised patient, but again, do not contain any live component of the virus, and the immunity that induced by this kind of vaccine is usually quite weak and require multiple booster dose. And one of the company vaccine candidate currently in phase 3 trial is Novavax. So after looking at a summary of the vaccine platform and few example, so I think all of you know this. About a week ago, our Prime Minister actually made a press statement and saying that he want to purchase a vaccine from this Pfizer, which is a messenger RNA vaccine. And Pfizer came out with the statement and say that after testing about 32,000 volunteers in a phase 3 trial, they found that their vaccine is 95% effective. And the efficacy of the vaccine was consistent across the age, gender, race, and ethnicity. And there is no serious side effect seen. So after this, Pfizer come up with this interim analysis, then all other vaccine candidate also very kiasu, and everybody come out and say that they are equally efficient like this Pfizer mRNA vaccine. So look at this Oxford University collaborate with the AstraZeneca company, the using adenovirus as a pseudovirus. So all of them require 2 doses of IM, 28 days apart, and they claim themselves have a 90% of efficacy, and they do not need an ultra cold temperature to keep the cold chain and low price. Moderna and Pfizer using messenger RNA, so they are quite similar, but Moderna claim that they do not need a so cold temperature to keep the vaccine safe. So the only issue with this Pfizer nowadays is just that not too sure that before the vaccine could have safely delivered to the user, whether or not this ultra cold condition could be maintained. This Subunit is a Russia vaccine using adenovirus as well, also claim themselves having 95% of efficacy in a low cost. Then from the China company, both using inactivated virus, they do not have any data for the efficacy yet. So actually all of these company and vaccine candidates are still having ongoing phase 3 trial. So I really think that at this moment, too early that we should draw a conclusion to say which one is better and which one we should try to use it. So regarding this COVID-19 vaccine, questions will remain more than answers. Even though all these companies trying to say that they are very good, more than 90% of efficacy, but actually until now it's still unclear that whether or not this vaccine could have blocked the transmission from asymptomatic people, then how well this vaccine is going to work for different groups of patients, immunocompromised patients and elderly. And if the vaccine is really working to prevent COVID-19 disease, but how long the immunity could have last? And we do not have the answer. We need to wait until to follow up the trials, the phase 3 trials. Only we know which one will be better.
Yeah, thanks, Apple. So I'm sure we have more questions later. We can write through our chat. And so if everyone is okay, then I'm going to share my screen and I'm going to present on my topic. Yep. So my topic is actually on current updates on of novel therapy, mainly on these 2 therapies, on monoclonal antibodies and JAK1/2 inhibitors. So thanks to Apple's so this is making my life much easier. But it's just in order to for everyone to understand the role of monoclonal antibodies and how does it work and everything. I think this slide is just making it in a very simplistic form and really helping me a lot. We know that vaccines work by teaching our body to recognize a foreign invader. In our case, it's actually a COVID-19, and thereby activates immune systems, go by our B cells and T helper cells and cytotoxic T cell activated. So, so whenever there's this activation of immune system, they produce antibody against the pathogens. what monoclonal antibody do is it's just taking a shortcut and skip all these steps, and therefore subsequently deliver a ready-made immune arsenal composed of identical manufactured antibody selected for maximum potency against SARS-CoV-2. Sorry. So monoclonal antibody, we know that it has to be produced from the plasma of a recovered patient. So different is that monoclonal antibody actually provides a singular activity against a predetermined target. It is genetically being DNA deciphered, and then it will be mass produced in the laboratory. And on the other hand, the convalescent plasma, as everyone knows, is actually more on the polyclonal antibody in the serum, which derive from patients who are convalescing from the infection. So theoretically, monoclonal antibodies potential benefits, number one, looking at the theories and everything into it, of course, it can help to provide immediate protection or treatment for those as exposed. And another way of it, potential benefit, is that it's something like a prevention option. Although it's short-lived, but prevention option before a vaccine is available. Or another way, sometimes it also can help with the benefit to people who actually cannot mount a very good, adequate immune response after a certain vaccination. So there were people talking about using a monoclonal antibody as a bridge for prevention and treatment before we even talking about letting the vaccines to come into pictures. But obviously, monoclonal antibodies, number one is availability because of the cost. And secondly, we understand that it's actually a short-lived protection also. So the idea of using monoclonal antibodies against infection actually is nothing new. In fact, it has been used, as everyone knows, for other viral infections such as influenza, RSV, MERS-CoV, and even Ebola virus. And if we really look into details, those that is actually effective in human trials are things like RSVs and Ebolas. So for COVID-19, as what our target, again, the spike protein. So the main target for our SARS-CoV neutralizing monoclonal antibodies is a surface spike glycoproteins, which mediate the viral entry into the host cells. So to be more specific, what they did is that the antibodies will block the S1 fragment of the SARS-CoV. The fragment of it, we call it as a receptor binding domain, and it's the ACE2 receptors. And by blocking into it, it can actually theoretically help to reduce the virus proliferation and disease severity. So in the month of November, there's a few announcements from FDA, if everyone's actually following the news. So these are the two companies. One is Eli Lilly, one is actually from Regeneron. In the month of November, FDA actually has been given an emergency use authorization for these two monoclonal antibodies. One is bamlanivimab. The other one, I was just gonna give a short name that's calling as Regeneron. It's a combinations of using they call it as a cocktail therapy. So mainly from if you really look into the FDA's updates, they are mainly is an emergency use authorizations are very specific, only can be used for those as non-hospitalized patients with mild to moderate COVID-19, who are at high risk of progressing to severe disease and hospitalization. So mainly the indication is for those non-hospitalized mild to moderate COVID-19 patients. And for this monoclonal antibodies, in fact, there has been a lot of FDA press release and everything. The only study that has really been published just recently is actually by this bamlanivimab. The other name is called LY-CoV555. Now they are naming it as a bamlanivimab, which is actually more of an interim analysis by Eli Lilly of an ongoing phase 2 randomized double-blind placebo-controlled trials. It's actually an outpatient trial whereby involving 450 patients of mild or moderate COVID-19 patients. So if you're looking into that, breaking into mainly actually 4 arms. One arm is of course placebo, and the other 3 arms are dividing into the different dosage of the monoclonal antibodies. So they are targeting mild to moderate patients, and most patients must be having a positive SARS-CoV-2 test within 3 days or within 3 days before infusion. What they are looking into in terms of endpoints are 2. Number 1 is looking into change of the viral load at day 11. And the secondary endpoint, of course, they'll look into the safety, the symptom severity, and more importantly, because this is an outpatient trial, they want to look into the rate of hospitalizations. So here, I just want to point out in terms of characteristics, so they are actually trying to also subcategorize and analyzing the 65 years and older, so-called higher risk age group, which constitute about 10 to 14% of the study participants. And the median durations of median days from the symptoms of onset is actually about 4 days, and most of them they have a CT value of about 23.8. And just summarize it into one slides about what kind of outcome did they come up. So primary outcome, when they look at the viral load, what they found is that at day 11 it was lower than that in the placebo group, only among those who received the 2,800 milligram dosing. So they don't find any benefits in 700 or 7,000 milligram, but in 2,800 milligram dose. And secondly, also they look into the hospitalizations. What they found is that the percentage of patients who were hospitalized when they are receiving placebo was much higher, around 6% compared to 1.6% in the treatment arms. So this is actually more on an interim analysis. I'm trying to see whether this difference of 6.3% and 1.6%, the difference, whether it is statistically significant, but I can't find any statistic about it. So I presume, I can't presume anything whether is it really significant or not. We really have to wait for the formal final report to come out. But what they did is that they also look into, they so-called also looking into the post-hoc analysis, whereby they focus on the high-risk group, i.e. those patients that is having more than 65 years old or having extreme obesity with BMI of 35. And the percentage of hospitalization is even much greater in this group, compared to placebo group with a treatment group. And also, they also found that from day 2 to day 6, so every day they're looking into the symptom scores, and they found that there is actually changes in terms of a symptom score from baseline, which is actually going into a advantage in the treatment group compared to the placebo group. So the thing is that when I read through this study, one thing I am concerned is that because the decrease of viral load at day 11, whether this is really a meaningful endpoint, especially from clinical point of view, we are not so very sure. And a lot of times, the viral load was actually, even in this study, what they found is that the viral load was already substantially reduced from the baseline for the majority of people, even including the patients at placebo group. So this finding was actually quite consistent with those, like, we are looking at like a natural cause of disease where the viral load will also come down by itself. So again, I would really think that we have to look at the final report to make a very, very good judgment on that. So what we're talking just now was actually an outpatient trial. Of course, subsequently also people will be more interested to look into any role of monoclonal antibody in the inpatient trial. So NIH is actually trying to chair the trial whereby they're actually combining this monoclonal antibody with remdesivir against a placebo and remdesivir group, and they run a phase 3 trial. Unfortunately, from the press release that released about 6 weeks ago, what they say is that they are recommending to pause the study because they don't find any clinical benefits in the bamlanivimab subgroup in the hospitalized patients. And so another company, the Regeneron company, this is the one of the cocktail that actually Donald Trump is taking, if everyone is actually wondering. So they actually also run trial, similar trial in the hospitalized patients. So what I do found from some of the press release, some of the FDA papers and things like they actually enroll the patient in the 4 independently randomized cohorts. Those at low flow oxygen, those that doesn't requiring oxygen at all, those on high flow oxygen, and those on mechanical ventilation. Again, from the news, what they mentioned is that they're actually suspending a trial and recruitment for those patients that is hospitalized which require high oxygen requirement. And what they document is that there's potential safety signal and an unfavorable risk-benefit profile in those patients that require high oxygen requirement. But we do not know much about the details, but we just kind of able to speculate that likely something doesn't look very right in this subgroup of patients, in this cohort of patients. And not, almost similarly within that couple of days, so they actually kind of released out some press release saying that Regeneron does work in those patients with this antibody cocktail, whereby they actually does work in those patients that is outpatient, which is a mild to moderate COVID-19. So it's a randomized, it's a placebo-controlled trial whereby they actually compare placebo with 2 different dosage of Regeneron, and mainly they are measuring in terms of viral load and the patient medical visits, i.e. The hospitalization, urgent care visits, and telemedicine visits. So all these things are actually never been able to be published. So what I am able to really look into detail is actually looking about the FDA report and look at the healthcare providers report, and they did actually give us some idea about what happens. And so the primary outcome there, which is a virological outcome, what they found is that in the outpatient trial, Regeneron does actually improve the viral load, especially better when if the patient having a higher baseline viral load, which actually derive a greater benefits. And in terms of the clinical results, of course, one of the most important things is that if you're outpatient, you were trying to look for indication for hospitalizations or any medically urgent visits. And they found that also there is some form of reduction in terms of COVID-19 related medical visits by almost half when they are through day 29. And the reductions of the medical visits actually even more pronounced if you are having 1 or more risk factors. You're having age group of more than 50 years old, BMI more than 30, immunocompromised state, or if you're having a lung, kidney, liver disease. So the table on the right is just looking at events on hospitalization or emergency visits. So it's a very raw table, so they don't provide any statistics, but what you can see is that roughly compare a placebo patient whereby 4% of them will attend to a hospital. Those with treatment arms is actually having a reductions of hospitalization by half, 2%. And so what do we know so far is that monoclonal antibody therapy for COVID-19, it does show some evidence of reduction of viral load and does show some reductions of medically attended visit in outpatients, mild to moderate COVID-19 patients. However, we don't find any benefits of treatments to be observed in patients that require hospitalization, especially those requiring oxygen therapy. In fact, for Regeneron, they actually they have stated that there's a potential worse clinical outcomes when administered to hospitalized patients with COVID-19 requiring high flow oxygen or mechanical ventilations. So as you can see, all these things, all this evidence I'm listing out, these are all not coming out from scientific papers, apart from the first one, which is also still an interim analysis. So I think we really need to have all these scientific papers being published, and then we look into it again. But roughly that's what happening for the past 1 month, and so far how's the evidence looks like. So this is in terms of how they look like. So in terms of monoclonal antibodies, those that is actually in a phase 3 trial, phase 2 or phase 3 is what I was just discussing. One is the Eli Lilly ones, and the other one is actually Regeneron's. There are also still others monoclonal antibodies, but they are all at a very, very early phase of the trial. But mainly you can see most of them are actually targeting again the spike proteins. So this is my first part of it, which is some updates about the monoclonal antibodies. My second part of my my presentation is actually talking a little bit about JAK inhibitors, more specifically actually baricitinib. So what we actually understand is that baricitinib is actually a JAK inhibitors, and it's actually used mainly in the rheumatology world, whereby they use it mainly for rheumatoid arthritis. But if you are using it for COVID-19, one of the main mechanism of action is that baricitinib is actually worked by fusion inhibitors, whereby they inhibits the fusion process of the viral entry into the host cell. And this baricitinib is actually having a high potential to bind to a protein kinase that regulate our viral endocytosis. So by inhibit viral endocytosis, that's what we're going to find proliferation. And at the same time, there is also a role in inhibition of cytokine release also. So this is where our baricitinib is actually going to work in terms of mechanism. In terms of USA, they're actually looking into, IDSA, they're actually looking into what kind of possible therapeutic that is under investigation. So baricitinib is actually one of the drug classes that is actually being labeled that is potentially going to be helpful, as what we have discussed in terms of mechanism of action. So specifically, if I look into some of the better paper that has been published for COVID-19 is by a group of Italian papers, but they are all actually retrospective and they are observational study. So what they did is actually in 7 Italian hospitals, they are looking into those moderate COVID-19 pneumonia patients, whereby they are actually compared 2 arms. One is actually a baricitinib Kaletra arm compared with a hydroxychloroquine Kaletra arm. So if you can see over here, looking from these retrospective studies, whereby they are actually aiming to look at mortality, their ICU admissions, and the discharge, they looks fairly promising. In fact, the baricitinib arms actually have reduction of death. In fact, there's no death in that arm. There's reduction of ICU admissions, and also there's a lot of higher rate of discharge also. And so it sounds very promising, but as you know, this is a retrospective. Eventually, they're going to tell you that you still have to do an RCT to confirm the findings and then to make it become a recommendation. So I quickly bring out this paper. The reason why I bring out this paper is that this is the Act 1 trial, whereby everyone knows that the remdesivir, the US group, whereby they actually published out the final report last month. Initially, in early years, whereby they actually published out interim report, and they are talking about whether remdesivir does reduce the time to recovery in hospitalized patients. In this paper, most of them are actually almost 75% are actually in a severe group of COVID-19, whereby they require oxygen. Yes, it does. From the paper, it does say that it actually reduced it from 15 days to 10 days. However, I think every one of us know that in terms of mortality, they can't prove it in this paper, and same thing in a solidarity trial paper later also, we can't prove that there is actually a significant mortality benefits in terms of reduced time to recovery. That's all. So one thing that the author was actually concluded is that remdesivir kind of helpful, but they are fairly aware that there is possibly given a high mortality despite the use of remdesivir. It is clear that treatment with antiviral drug alone is not likely to be sufficient. And therefore, they believe that there must be some future strategies looking into a combination therapy. And one of it that they are talking about is actually the ACTT2 trial, whereby they are combining baricitinib with remdesivir. So that is all these things combined. In fact, I think most of the scientists over there, they're actually believing that a combination of antiviral therapy using a different mechanism of action does work better. And there are some Japanese papers also talking about it. And similar on the same month on November 19, so FDA again actually issued an emergency use authorizations for a so-called this baricitinib combination therapy, and so they are mainly indicated for this group of patients. As of yesterday, I still can. These ACTT trial papers are not published yet. So as what I've just mentioned for earlier on, I'm actually getting most of the information from the FDA report for the healthcare worker. But essentially, this ACTT2 trial, as what I mentioned, is actually a randomized double-blind placebo-controlled trial, whereby you're evaluating the combination therapy of baricitinib and remdesivir compared to remdesivir alone. So we are looking into a fairly ill populations, whereby patients must be confirmed COVID-19 with radiological imaging, or the patient is hypoxic with SpO2 less than 94%, requiring supplemental oxygen, or if the patient is using ECMO or mechanical ventilations. So they're looking into 1,000 patients of moderate to severe COVID-19. They divide it half into a treatment arm and the other half into a placebo arm, which is a placebo plus remdesivir arm. So the outcomes they're looking into it is a little bit by like ACTT trial, whereby the primary outcome they look into time to recovery within 29 days. And the key secondary outcomes was actually more on the clinical status on the day 15, based on the 8-point ordinal scales. And I'm sure everyone is very familiar with this 8-point ordinal scale. It has been used during the early trial also, whereby a scale of 8 or 7 or 8, that means it's even more severe, like either hospitalized with ventilation or death. And 1 and 2 is those as ambulatory, and 2 is ambulatory and with some limitation of activities. And yeah, I'm not going to list down everything. So so some of the information that I can get, I just want to share it with everyone, at least as some idea about it. The mean age of the paper for those in these participants is actually 55 years old, with 30% of the patients aged 65 years and above. 63% of them were male, and if you can look into the breakdown in terms of the severity of it, almost about 87% of them actually are requiring some form of oxygen, which means that this population of patients are, for us, is already a severe COVID, at least stage 4 kind of COVID-19 patients. Their common comorbidities is actually obesity, hypertension, and type 2 diabetes, and the demographic and disease characteristics were balanced between the two groups. So as I mentioned, these are the key results that they actually kind of putting up in the FDA papers. They found that in terms of median time to recovery, those in the baricitinib arm has 1 day earlier recovery compared to a placebo arm, which is statistically significant. In terms of clinical status on day 15, again, the treatment arms was saying that it's more likely to have a better clinical status at day 15 compared to a placebo arms. And proportion of patients who die or progress to NIV or mechanical ventilation by day 29 was also lower in the baricitinib arm compared to the placebo arm. And lastly, when we look at the proportion of patients who die by day 29, they found that the baricitinib arm has a trend towards a lower mortality. Of course, if I look at when I look into the 95% confidence interval, they don't put a p-value, but if you look into it, it looks like it's possibly not statistically significant, but certainly it looks like there is a trend towards benefits in terms of mortality when you look into baricitinib arm. So this is what this paper is about. And in terms of so-called safety, compared to a placebo groups, mainly they don't see any difference in terms of grade 3 or grade 4 or serious adverse effects. So was reported as fairly safe. So just one slide. When we talk about JAK1, JAK2 inhibitors, another one JAK inhibitors that people were talking about is a ruxolitinib, which is actually one of the JAK inhibitors that has been used more extensively in the hematology field. They use it for myelofibrosis, PCV, steroid-resistant GVHD, and things like that. So so I think early on, when in China, the time they actually did a pilot studies whereby they are actually randomized it 1-to-1 towards ruxolitinib and placebo, hoping that actually they are actually coming with the same kind of findings. So the primary, they are looking into just a pilot study, so it's only 20 patients compared with another 20 patients of ruxo, looking into those patients that are having a severe COVID-19. Of course, their endpoint is to looking into their improvement rate, clinical improvement rate, the incidence of death, and the viral clearance. And very quickly, you can see that all these outcomes are actually not significant. They can't find any significance about ruxolitinib. That's why I think subsequently ruxolitinib was not being suggested as one of the potential treatment for COVID-19. So if you want to talk about JAK inhibitors for COVID-19, what I do understand is that number one, use baricitinib as a monotherapy sounds okay, with some retrospective studies showing some promise, very promising, but it has not been evaluated in the randomized studies. So we do not know for sure. And it sounds like from this ACTT trial, 2 trial, there is a role of combination therapy, and there are few statistically significant benefits in terms of clinical recovery, in terms of improvement of clinical status. But does it translate against one trial outcomes will later translate into many more questions that we're going to answer. Does it translate the same efficacy for other antiviral like favipiravir with baricitinib and other on? We don't know. And in order to further really scrutinize whether that paper, how good is it, we probably have to really wait for the formal publication of the scientific papers. Yeah, and with that, actually, that's my updates for these two therapies. Thank you very much. Yeah. Happy to get questions. Yeah.